Hello viewers, welcome to the session on food technology. Today's topic is fat-soluble vitamins. Vitamins are a group of organic compounds that are required in very small amounts, essential for the normal functioning of the human body. They have widely varying chemical and physiological functions and are broadly distributed in natural food sources. 13 vitamins are recognized in human nutrition, and these may be conveniently classified into two groups according to their solubility. The fat soluble vitamins are vitamins A, D, E and K, and the water soluble vitamins comprise vitamin C and the members of the vitamin B group, namely thiamine or vitamin B1, riboflavin or vitamin B2, niacin, vitamin B6, pantothenic acid, folate and vitamin B12. To learn more about vitamins, their absorption, transport and functions, the session is divided into 5 major topics, namely transport and absorption of vitamins, fat-soluble vitamins, functions and their forms, structure of fat-soluble vitamins, stability of fat-soluble vitamins, and bioavailability of fat-soluble vitamins. Firstly, transport and absorption of vitamins. Absorption is a complex process in which the nutrients pass through the intestinal mucosal cells into the bloodstream. Mammalian epithelia are enveloped by a plasma membrane composed of a phospholipid bilayer interspersed frequently with cholesterol molecules. Integral transmembrane proteins span the lipid bilayer in a weaving fashion and account for most membrane associated receptors, transporters and certain enzymes. Tight junctions prevent the passage of water and molecular solutes between adjacent epithelial cells. The lipid bilayer along with membrane associated receptors is displayed on the screen. The plasma membrane constitutes a selective barrier to the transcellular movement of molecules and ions between the extracellular and intracellular fluid compartments. Fat-soluble substances, water and small uncharged polar solutes can simply diffuse through the membrane, but ions and water-soluble molecules having 5 or more carbon atoms cannot do so. Most biologically important water-soluble substances such as glucose, amino acids, water-soluble vitamins and certain inorganic ions are translocated across the plasma membrane by means of protein transporters, which exert their effect through a change in their three-dimensional shape. Specific transporters are responsible for the translocation of a specific molecule or a group of closely related molecules. Specificity is imparted by the tertiary and quaternary structures of the transporter molecule only if a solute's spatial configuration fits into the protein will the solute be transferred across the membrane. Transporters fall into two main classes, carriers and ion channels. Ion pumps are a type of carrier protein which is also an enzyme. At physiological concentrations, the translocation of several water-soluble vitamins, that is thiamine, riboflavin, pantothenic acid, biotin and vitamin C across the cell membranes is mediated by carrier proteins as displayed on the screen. The term transport implies a carrier-mediated translocation. The downhill movement of a substance from a region of higher concentration to lower concentration is a passive process driven by the concentration gradient. There are two types of passive movement: facilitated diffusion, which is carrier mediated, and simple diffusion, which is not. The uphill movement of a substance, that is movement from lower to higher concentration gradient, is referred to as active transport and requires the expenditure of metabolic energy. Primary active transport is driven directly by metabolic energy and is carried out extensively by ion pumps such as the calcium pumps, the sodium pump and the proton pumps. Ion pumps are ATPases which utilize the energy released by the hydrolysis of ATP. Secondary active transport is indirectly linked to metabolic energy through a coupling of the solute to the pump driven movement inorganic ion, usually sodium ion. Intestinal absorption. The functional absorptive unit of the small intestine is the villus, a finger-like projection of the mucosa. The lamina propria core of each villus contains a capillary network with a supplying arteriole and draining venule. Each villus is covered by an epithelium composed of single layer of columnar absorptive cells called enterocytes, interspersed occasionally with mucus secreting goblet cells. The enterocytes constitute the only anatomical barrier of physiological significance controlling the absorption of nutrients. The apical membrane of the enterocytes, that is the membrane facing the intestinal lumen, is covered with microvilli, which are minute projections of the plasma membrane. Because of its brush-like appearance under the microscope, the apical membrane is also known as the brush border membrane. The picture of villus is displayed on the screen. Absorption of fat-soluble vitamins. Absorption of the fat-soluble vitamins takes place mainly in the proximal jejunum and depends on the proper functioning of the digestion and absorption of the dietary fat. The stomach is the major site of emulsification of fat. The coarse lipid emulsion on entering the duodenum is emulsified into smaller globules by a detergent action of bile. Pancreatic lipase hydrolyzes triglycerides at the 1 and 3 position, yielding 2 monoglycerides and free fatty acids. During their detergent action, bile salts exist as individual molecules. Above a critical concentration of bile salts, the bile constituents, that is bile salts, phospholipids and cholesterol, form aggregates called micelles, in which the polar end of the molecule are oriented towards the surface and the non-polar portion forms the interior. The picture of micelles is displayed on the screen. The 2 monoglycerides and free fatty acids are sufficiently polar to combine with the micelles to form mixed micelles. These are stable water-soluble structure which can dissolve fat-soluble vitamins and other hydrophobic compounds in their oily interior. Mixed micelles do not cross the brush border membrane of enterocytes as intact structure. The products of lipolysis must dissociate from these structure before they can be absorbed. Individual lipids including fat-soluble vitamins can then be passively be absorbed across the brush border membranes. After the lipolytic micellar product enters the enterocytes, a cytosolic fatty acid binding protein facilitates intercellular transport of fatty acids by directing them from the cell membrane to the smooth endoplasmic reticulum where triglyceride synthesis takes place. The triglycerides are packed into chylomicrons together with free and esterified cholesterol, phospholipids, apolipoprotein, fat-soluble vitamins and carotenoids. After further processing, the chylomicrons are discharged from the enterocytes by the exocytosis across the basolateral membrane and enter the central lacteal of the villus. From there, they pass into the larger lymphatic channels draining the intestine into the thoracic duct and ultimately into the systemic circulation. Associated with the endothelium of blood capillaries in most tissues is the enzyme lipoprotein lipase, which attacks circulating chylomicrons and convert them into smaller triglyceride depleted particles known as chylomicron remnants. These particles contain apolipoprotein E or ApoE acquired from other circulating lipoproteins. The released free fatty acids and diglycerides can then be absorbed by the tissue cells. The liver has the capacity to rapidly remove chylomicron remnants from the circulation. Fat-soluble vitamins, functions and their forms. Vitamin A, D, E and K are regarded as fat-soluble vitamins. They are stored in the liver and require bile salts for absorption. Vitamin A. Vitamin A active compounds include retinoids designated as vitamin A and their carotenoid precursors, that is provitamin A carotenoids. The retinoids comprise retinol, retinaldehyde and retinoic acid together with their naturally occurring and synthetic analogues. Retinal and retinoic acid are formed from retinol. Further, retinal and retinol are interconvertible. But the retinoic acid cannot be converted either to retinal or retinol. In plant foods, it is present in provitamin form, which is known as carotenes. There are three types of carotenes present in plants. They are alpha carotene, beta carotene and gamma carotene. Beta carotene are most potent source of retinol because one molecule of beta carotene yields 2 molecules of vitamin A in vivo. Generally speaking, dietary vitamin A is obtained from animal derived foods, while plant foods provide carotenoid precursors such as beta carotene, beta cryptoxanthin, zeta carotene, xenoxanthin, zeaxanthin, neoxanthin and violaxanthin. Vision is a non-hormonal biochemical process involving a different vitamin A metabolite. Vitamin A is an essential dietary factor for normal embryogenesis, cell growth and differentiation, reproduction, maintenance of the immune system and vision. Retinol is required for differentiation and function as steroid hormone. Retinol and retinoic acid are involved in regulation of gene expression. Vitamin A is required for the maintenance of nervous tissue, particularly myelin sheath formation. Vitamin D. Vitamin D is represented by cholecalciferol, that is vitamin D3, and ergocalciferol, that is vitamin D2, which is structurally similar corticosteroids derived from the UV irradiation of provitamin D sterols. Exposure of the skin to sunlight converts 7-dehydrocholesterol to vitamin D3, which on reaching the blood capillaries of the dermis is conveyed to the liver on a specific plasma transport protein. Vitamin D2 is produced in plants, fungi and yeast by the solar irradiation of ergosterol. On irradiation, the provitamins are initially converted to pre-vitamin D, which undergoes thermal transformation to vitamin D. Vitamin D in its active form 1,25-dihydroxycholecalciferol or calcitriol act as steroid hormone. It is synthesized from ergocalciferol and cholecalciferol. Vitamin D binds to chromatin of target tissue and expresses the genes for calcium binding protein and calcium ATPase in the intestinal cells, which increases calcium absorption by actively transporting calcium ions across the plasma membrane. It is essential for mineralization of bones by stimulating the transcription of mRNA for calcium binding proteins and alkaline phosphatase, thus increasing the absorption of calcium and phosphate ions in bone mineralization. Vitamin D decreases the pH of the lower gastrointestinal tract. It helps in the excretion of phosphate and citric acid. Calcitriol is an immunoregulatory hormone. It stimulates cell-mediated immunity. It plays a vital role in monocyte or macrophage activation. Vitamin E. The term vitamin E refers to a group of 4, often 6 compounds that exhibit vitamin E activity. They are alpha tocopherol, beta tocopherol, gamma tocopherol and delta tocopherol. Alpha tocopherol present in cell membrane of subcellular organelles and in cytosol function as antioxidants or free radical scavengers. It is present in high concentration in tissues which are exposed to high oxygen pressure like erythrocytes, lungs and retina. It prevents peroxidation of membrane lipids, particularly polyunsaturated fatty acids of membrane phospholipids. Vitamin E is required for fertility in animals. Vitamin E increases synthesis of heme proteins by increasing synthesis of ALA synthetase and ALA dehydratase. Vitamin E prevents dietary vitamin A and carotenes from oxidative damage. Vitamin K. The term vitamin K refers to a group of compounds that exhibit vitamin K activity. They are vitamin K1, also known as phylloquinone, is the major form of vitamin found in plants, particularly in green leafy vegetables. Vitamin K2, also known as menaquinone, is the vitamin K present in animals and synthesized by intestinal flora. Menadione is the synthetic analog of vitamin K. It is also called as vitamin K3. It lacks characteristic side chain present in vitamin K1 and K2. It is converted to vitamin K2 by alkylation in the body. Vitamin K is essential for the activation of specific proteins such as prothrombin involved in blood clotting and in bone mineralization through its role as a cofactor for gamma glutamyl carboxylase, gamma carboxyglutamate residue, allowing the protein to bind calcium and thus become activated. Structure of fat soluble vitamins. Vitamin A. The parent vitamin A compound retinol has the empirical formula C20H30O. The molecule comprises of a beta ionone or the cyclohexanyl ring attached at the C6 position and a side chain containing 2 isoprene units with 4 conjugated double bonds, which give rise to cis-trans geometric isomers. Carotenoids can be considered chemically as derivatives of lycopene, a C40H56 polyene composed of 8 isoprenoid units. Derivatives are formed by a variety of reactions that include cyclization, hydrogenation, dehydrogenation and insertion of oxygen. Hydrocarbon carotenoids are known as carotene and the oxygenated derivatives are termed as xanthophylls. In nature, carotenoids exist primarily in the all-trans configuration. The structure of vitamin A is displayed on the screen. Vitamin D. All forms of vitamin D belong to a family of lipids called seichosteroids. Seichosteroids are very similar in structure to steroids except that 2 of the B ring carbon atoms of the typical 4 steroid rings are not joined. The structural difference between vitamin D2 and vitamin D3 is the side chain of D2 contains a double bond between carbon 22 and 23 and a methyl group on carbon 24. The structure of vitamin D is displayed on the screen. Vitamin E. Tocopherol are methyl substituted derivatives of tocol which comprise of a chromanol ring attached at C2 to a saturated isoprenoid side chain. Tocotrienols are analogous structures whose side chains contain 3 trans double bonds. In nature, there are 4 tocopherols and 4 corresponding tocotrienols. These are designated as alpha, beta, gamma and delta according to the number and position of the methyl substitutes in the chromanol ring. They differ in methyl groups in position 5, 7 and 8 of chromanol ring. Alpha tocopherol has 3 methyl groups in positions 5, 7 and 8 of chromanol ring. The chromanol ring of beta and gamma tocopherols contain 2 methyl groups in 5, 8 and 7, 8 respectively. However, gamma tocopherol has 1 methyl group in position 8 of chromanol ring. The structure of vitamin E is displayed on the screen. Vitamin K. They are derivatives of naphthoquinone and differ in side chain. Phylloquinone contain phytyl side chain, whereas menaquinone contains polyisoprenoid side chain made up of 7 isoprene units. Several variants of vitamin K2 containing more than 7 isoprenoid units in the side chain are also identified. The structure of vitamin K is displayed on the screen. Stability of fat soluble vitamins. Vitamin A. Retinol is readily oxidized by atmospheric oxygen, resulting in an almost complete loss of biological activity. The 5,6-epoxide and 5,8-furanoxide are among the oxidation products. Retinyl esters are more stable towards oxidation when compared to retinol. Retinol is extremely sensitive towards acids, which can cause rearrangement of double bonds and dehydration. Solutions of all trans retinol or retinyl palmitate in hexane undergo slow isomerization to the lower potency cis isomers when exposed to white light. The photoisomerization rate is greatly increased in the presence of chlorinated solvents. However, it is stable in the dark. Irradiation also rearranges double bonds to form inactive retrostructures. The carotenoids are stable within their natural plant cell environment, but once isolated, they are prone to molecular rearrangement, trans to cis isomerization, and degradation by heat, light, oxygen, trace amounts of acids, and active surfaces such as silica. They are sensitive to enzymatic and non-enzymatic oxidation. The enzyme lipoxygenase in plant tissue catalyzes lipid peroxidation, giving rise to hydroperoxides. The hydroperoxides decompose to form peroxyl and alkoxyl radicals, which attack carotenoids. The cutting of fruits and vegetables into small pieces or maceration increases the exposure to oxygen and brings the carotenoids and enzymes together. Moderate heat treatments such as blanching and cooking denature carotenoid binding proteins, thereby releasing the carotenoids so that they can be more readily extracted. Blanching of fruits and vegetables before processing inactivates lipoxygenase and other enzymes, that is peroxidase, that are involved in carotenoid destruction. Vitamin D. It is sensitive to oxygen and light. In the presence of mild acid, vitamin D undergoes isomerization. Vitamin E. Tocopherols are alkaline sensitive and their vitamin activity is destroyed by oxidation. Cooking and food processing may destroy vitamin E to some extent. Tocopherols and tocotrienols are destroyed fairly rapidly by sunlight and artificial light containing wavelengths in the UV region. The vitamins are slowly oxidized by atmospheric oxygen to form mainly biologically inactive quinones. The oxidation is accelerated by light, heat, alkalinity and certain trace metals. The tocotrienols, by virtue of their unsaturated side chain, are more susceptible to destruction than the tocopherols. Vitamin E is the most radiation sensitive of the fat soluble vitamins. Presence of lipoxygenase destructs the vitamin. Vitamin K. Compounds of vitamin K are decomposed by UV radiation, alkali, strong acids and reducing agents, but are reasonably stable to oxidizing conditions and heat. Phylloquinone is sensitive to both fluorescent light and sunlight. However, they are stable to cooking, gamma irradiation and freezing. Bioavailability of fat soluble vitamins. Bioaccessibility is defined as the fraction of vitamin transferred during digestion from the food matrix to mixed micelles and thus made accessible for absorption. whereas bioefficacy defines the percentage efficiency with which ingested vitamin is absorbed and converted to its active form in the body. When meals containing natural amounts of vitamin A and provitamin A carotenoids are consumed, vitamin A is absorbed with an efficiency of 70 to 90% compared to 20 to 50% of the provitamins. Bioavailability of carotenoids is frequently assessed by absorption efficiency, which is defined as the percentage of ingested carotenoid that is secreted into the general circulation and therefore made available for tissue uptake. Bioavailability also depends on certain nutrients. The serum level of vitamin A is lesser in subjects with severe protein malnutrition. Protein is necessary for the mobilization of vitamin A from the liver to the bloodstream. Dietary fat is also important for micelle formation, thus facilitating the absorption of hydrophobic carotenes. Dietary fiber inhibits the absorption of lipids and therefore the absorption of fat-soluble vitamins. Vitamin D given with milk has been reported to be 3 to 10 times more potent than given with oil, the stimulatory factor being attributable to the lactalbumin fraction. Alcoholism and smoking reduces the serum level of vitamin D. Plant sterols and PUFAs decrease the absorption of vitamin E. The efficiency of vitamin K absorption varies widely depending on the source of the vitamin and the amount of fat in the meal. Presence of fat enhances absorption. The tight binding of phyloquinones to the thylakoid membrane of the chloroplast explains the poor bioavailability of the vitamin in green plants. The free phyloquinone in vegetable oils, margarine and dairy products is well absorbed owing to the stimulating effect of fat. To conclude, translocation of nutrients occurs between membranes by different processes such as active transport and passive diffusion. The transport system varies according to the nature and solubility of nutrients. The transport of fat-soluble vitamins mainly requires chylomicrons which carry them to the target tissue. Fat-soluble vitamins are hydrophobic molecules and isoprene derivatives. The bioavailability of fat-soluble vitamins is affected by high intake of fiber, smoking and alcoholism. Thank you.